In this episode, we'll explain why careful standardization of nomenclature, removal of duplicate records, and correction of obvious errors are essential for accurate interpretation of vegetation data. Our example comes from a dataset on semi-natural forest vegetation in the Mohelnička Valley, located in the southeastern part of the Czech Republic. The vegetation records were collected in the field and digitized between 2008 and 2025 by 120 students of the Geobotany field course at Masaryk University, under the supervision of 6 experienced university teachers. The result is a data set of high didactic value that, without proper cleaning, may easily lead to misleading interpretations.
In the dataset, each row, each taxon carries the same weight. This means that if we enter the same species under different taxonomic concepts, such as Veronica chamedris under an outdated name, Veronica vindobonensis as the correctly identified flowering plant, or a broader aggregate name when recorded as a seedling, these entries will artificially increase dissimilarity between plots recorded in different years. Yet, in all cases, they refer to the same biological taxon.
We also need to remove obvious errors caused by data entry oversight. A common mistake, for instance, is misclassifying woody species as part of the herb layer. Because species are recorded using abbreviations, typos are also quite frequent. These types of errors introduce unnecessary additional noise into the dataset.
Make sure the records are internally consistent. In our dataset, for example, bryophytes and lichens were consistently recorded during the early years, but this practice was later abandoned. To ensure comparability, the dataset must be standardized, meaning bryophytes and lichens should be removed. Otherwise, their presence would distort patterns and compromise the validity of later analyses.
Undetermined taxa, often poorly identifiable seedlings, are typically recorded only at the genus level. While these records are important for species counts, or when publishing complete tables, they add noise and burden to data analyses, especially in classification and ordination. The genus in question is usually represented by multiple species in the dataset, and we cannot reliably match the unidentified record to any of them. In such cases, it's better to exclude these ambiguous entries before running analyses.
Trees and shrubs are often recorded in multiple vegetation layers simultaneously. This gives such species greater weight and apparent importance in the dataset. However, in European vegetation, woody species typically occupy broad ecological niches and therefore have relatively low indicator value compared to other species. For this reason, trees and shrubs recorded in multiple layers are often merged into a single entry before analysis, to ensure they carry the same weight as other species in the dataset. In some cases, for the very same reason, it may be more appropriate to work with presence-absence data only.
Previous examples show properly preparing vegetation data to yield reliable and interpretable results, independent of who collected them, is one of the most demanding and expertise-intensive tasks in vegetation science. The original unprocessed dataset contained 277 taxa across 60 plots. After data cleaning and standardization, the number of taxa dropped to 139. We'll demonstrate the impact of this transformation using an ordination diagram produced by the non-metric multidimensional scaling method. The unprocessed data reveal a generally correct underlying pattern. The first axis reflects increasing moisture and nutrients, while the second axis corresponds to increasing temperature requirements. However, the vegetation records of scree forests located at the valley bottom show an illogically high spread, almost as wide as that of thermophilous and acidophilous oak forests found on the opposite sun-exposed slopes. After applying all nomenclatural corrections, removing errors, merging vegetation layers, and using an appropriate Hellinger transformation, which normalizes species cover and downweights dominant species, the dataset reveals a much more interpretable pattern. Scree forests now show only negligible differences, while oak hornbeam forests from the central parts of the northern and southern slopes display moderate variation. In contrast, thermophilous and acidophilous oak forests on the upper northern and southern edges of the valley form clearly distinct communities.
That's why it's important to be very careful when preparing your vegetation data for analysis. A carefully prepared dataset reflects the skill and professionalism of the vegetation scientist. In the following chapters, we'll explain the technical steps you can take to make sure your dataset is clean, consistent, and ready for interpretation.